I really appreciate the opportunity to talk today and to chair this session. It's a really exciting session. Metabolism, neurometabolism is something that's dear to my heart and something I think that's very important when we look at kiddos with autism and other neurodevelopmental disorders. One of the most important things I think about metabolism and what makes me excited about it is that it gives us a target to be able to change how the way the physiology of the body is working once we understand it. And I think that we're understanding more and more. And today we have met some really just excellent speakers, most of them and my colleagues that I've worked with who I think are just really spearheading this research forward in incredible ways and will help us understand autism and neurodevelopmental disorders and what we can do about it. I'll introduce them as we go along. First, I will start with my talks. I have two talks today to talk about. The first one, I will talk about and pull up right away. And then we'll go on to the other talks following that to hear about other exciting work. Let's see. I think Pramila, have you given me am I the host?
Yes. What?
Okay.
Yeah, just start screen share.
Okay. I'm sorry. There we go. So can you see my presentation?
Yes.
Good. Excellent. Super. Little hiccup with Zoom. So today, my first talk, I'll talk about the prenatal environmental influences on autism symptomatology and how that actually is seems to be mediated, we think, through the mitochondria. This is some exciting new research that we've just published recently. My disclosures: I have funding from various organizations. We run certain industry clinical trials, and I'm on the advisory board of several companies. Disclaimer, I always like to say, we talk about treatments here sometimes, and these are most of the time, they're not FDA approved treatments. They're off-label treatments, which there's nothing wrong with. But it means that really any type of treatment, really you would need to consult with an expert in the field regarding such treatments. So mitochondrial disorders seem to be somewhat prevalent in autism. And I'd just like to go back to this meta-analysis and systematic review that Dr. Rosignol and I wrote back in 2012. And this is when we started to realize that there is a lot of literature suggesting that there may have been some mitochondrial disorders associated with autism. So we went through and reviewed all the papers, the literature, and we found something interesting. And here we can see that one of the parts of the paper we reviewed is we looked at every case of mitochondrial disease and autism to see how it differed from the diagnosis of general ASD and mitochondrial disease in general. And we found some really interesting things. We found that kiddos with mitochondrial disease and autism tended to more commonly have developmental regression, seizures, motor delays, and GI abnormalities greater than what we see in the run-of-the-mill autism. But One of the really interesting things is that only 23% of the cases of mitochondrial disease in autism could there really be a genetic abnormality found to explain the autism. I'm sorry, explain the mitochondrial disease. So, that really suggested, well, maybe the mitochondrial disease was not due to a genetic disorder. There may be other factors that may be making the mitochondria not work correctly. And I think the first paper that's really kind of hinted that there might be a different type of mitochondrial dysfunction associated with autism is really this paper in 2000 published in the Journal of Child Neurology by Graf and co-authored by Dr. Navio, who'll be speaking later. And it is really an interesting paper because what it showed is that the same genetic mutation that caused Leigh syndrome in a sibling, in the female sibling of the sibling pair, caused autism in the boy. So the girl sibling had Leigh syndrome, and which is a known mitochondrial disease, but the boy had autism. And when they looked at how the mitochondria was functioning, the girl's mitochondria, which is more classic for mitochondrial disease, showed that it was not working very well. Complex 4 and 5 were very low. They were functioning very poorly. But for the boy, it was the opposite. They looked at it and complex 1 was actually functioning above normal, very, very had a high activity, which is something that really is not always recognized when we think of mitochondrial dysfunction. And I think, people say, okay, well, if the mitochondria is working really well, what's the significance of that? And we'll see in the future. So I think then one of the second papers that really showed us that this pattern may be somewhat linked to autism was this paper that Dr. Navio and I published. And this is when I started working up kids with autism. We would do a muscle biopsy if they had signs of mitochondrial disease. And what we started finding is that rather than the electron transport chain showing very low function, that we had a series of kids who had complex 4 that was very, very high. It was 200% of normal. So they had signs of mitochondrial dysfunction. They actually had electron microscopy changes, some of them. And instead of their mitochondria working very poorly, as we see in classic mitochondrial disease, it was working, it was overactive. And it ends up that we're not the only ones that reported this. When you look in the literature, that there another report that looked at brain tissue from individuals with autism actually showed that complex 4 was working overtime, or it was much higher than that of controls. And of course, we wanted to study this a little bit more, and you can't do muscle biopsies or brain biopsies or such on a lot of kids with autism. It's just not practical. But what you could do is you can use something called buccal swab technique, where you take some of the cells from inside of the cheek and you send them to the laboratory. In this case, this is Dr. Goldenthal's laboratory, and he's the one that developed this test to look at enzymatic function. In this picture here, we see that we measured citrate synthase, which is a Krebs cycle or a citric acid cycle enzyme, and tells us about overall mitochondrial content, complex 1 activity and complex 4 activity. And the black dots are the ones that are within normal range. The red are the ones that are within or outside normal range, either low or high. And we see that kids with autism citrate synthase, some were below normal function, but many were very high. For complex 1, it was the same thing. We had many that had below normal complex 1 function, but also several with several high function. And for complex 4, we found several that had elevated complex 4 activity. And this really goes to this point where we think that, well, if we look at autism, it overlaps with classic mitochondrial disease, which we divide into definite, probable, and possible mitochondrial disease. But it seems to overlap more with this non-classical mitochondrial disease, or what we call mitochondrial dysfunction. And so one way to look at this, we were very interested in the mitochondria and why it may not be functioning properly. So one of the things we knew is that it seems that kids with autism seems to have high levels of oxidative stress. And this is something that can disturb mitochondrial function. So there's high levels of oxidative stress and reactive oxygen species. It actually can affect the mitochondria. So one of the things we did in the laboratory is we used a device called the Seahorse, which is a high throughput respirometer, which measures mitochondrial function in living tissue. And in this case, we used lymphoblastoid cell lines from kiddos with autism. And we compare them to control cell lines from matched from patients of similar age and gender. And this assay gives us 3 measures, which I'll talk about. It gives us a measure of basal respiration, of how the mitochondria are kind of working at baseline. ATP-linked respiration, how much ATP the mitochondria is making. Proton leak, which tells us about about how the mitochondria is handling oxidative stress at the inner mitochondrial membrane. Maximal reserve capacity, or maximal respiratory capacity, and that's how hard the mitochondria can work if it had to. And reserve capacity, which is a very important number because we know that when the reserve of the mitochondria falls down below 0, it means the mitochondria is not working well. And many times we think it's signaling the cell for apoptosis. So the first thing we did is we compared these groups of cell lines, and then on this in the Seahorse, and we also used something called DMNQ to increase oxidative stress in the laboratory in the cell lines for about an hour before we did the assay to see how the mitochondria respond to oxidative stress or increases in oxidative stress. So so when we look at the baseline, and baseline mitochondrial function would be at DMNQ0, what we found is, to our surprise, is that the mitochondria in the kiddos with autism, which are labeled AD here, were actually working above normal. That is, ATP-linked respiration was above the controls, and maximal respiratory capacity was above the controls, and reserve capacity was above the controls. So again, instead of working very poorly, as you would see in mitochondrial disease, they actually seem to have increased respiration. But then what we saw is that when we stress them by increasing DMNQ, these numbers actually fell back towards normal, and in the case of reserve capacity, below normal, suggesting that just a low amount of reactive oxygen species caused the mitochondria to lose its reserve capacity significantly. Of course, everything in autism is subgroups, so we used a cluster analysis to see if there was a subgroup driving this, and we found that indeed there was, and we call these ADAs for autistic disorder cell lines with abnormal mitochondrial function. And then when we graphed them out, we looked at these parameters in the ADAs, these cell lines that seem to have abnormal mitochondrial function. The data was striking. And so this is the middle row here in this graph. And we could see that ATP-linked respiration, maximal respiratory capacity, and reserve capacity was twice that in the cell lines with abnormal mitochondrial function as compared to controls. And as we stress these cells, we increase DMNQ, we see that the reserve capacity dropped very quickly and precipitously went below control, suggesting a sensitivity to reactive oxygen species. Again, we're interested in the overlap between reactive oxygen species and how it affects mitochondria. And given the fact that we know that siblings of kiddos with autism seem to also have some markers of oxidative stress, we use this to pull out whether it was specific to the mitochondria or to oxidative stress that was really related to the autism. That is that we found siblings, we got sibling cell lines, and they had similar redox abnormalities. And then we asked, well, did they have similar mitochondrial abnormalities? And what we found is that they didn't. Even though the siblings had markers of increased oxidative stress and that their mitochondrial profile overlapped that of controls. And it was the kiddos with autism that had this abnormal mitochondrial profile with increased mitochondrial respiration. Why is this occurring? You know, we really have many hypotheses, but we think that in some ways, you know, you can set up a vicious cycle where the mitochondria increase the amount of oxidative stress in the cell, and that causes some mitochondrial dysfunction, and you can end up having a vicious cycle. And here's just one example of how that might work. That is that to make glutathione, which is the major antioxidant, you need to have ATP to make it de novo, and glutathione is the major antioxidant which controls oxidative stress in the mitochondria. So if the mitochondria isn't working, then you're not going to make glutathione. Or if you need more glutathione, you may need to have the mitochondria work overtime to make ATP, which is a possibility. So some of the things that we did find when we looked at these cells in the laboratory, these abnormally functioning lymphoblastoid cell lines, is that they had upregulated a protein called UCP2, which controls oxidative stress at the inner mitochondrial membrane. And that's consistent with an increase in proton leak that we see in the seahorse. And then interestingly, some more compelling data is we looked at the expression of certain genes and that were important for regulation of physiological stress in the face that are involved in mitochondrial, involved in helping the mitochondria actually adapt to oxidative stress. And what we found was something interesting. Now, both of these cell lines, the ADAs and the ADNs, have markers of increased oxidative stress, and so we figured that all of these genes that respond to oxidative stress and physiological stress related to the mitochondria would be increased. But we found that wasn't the case. We actually found that only the cells that had normal mitochondrial function were upregulating these stresses, these genes that were related to really control oxidative stress and allow the mitochondria to adapt. And what we noticed, so those are all in green. So the ones in green are, are these genes that are involved in in helping the mitochondria to handle oxidative stress and physiological stress. And they were only increased in the ADAs. And what we noticed, though, is that these were inhibited by a arm of the pathway downstream from mTOR called the S6K1, which is inhibited by low-dose rapamycin. So what we did is we did a study where we actually used low-dose rapamycin, and we showed that if you use low-dose rapamycin, you actually can normalize the respiratory rates of these cells. That is, on the top, you can see the ADAs. With the rapamycin, we actually decreased these respiratory rates. And then we correlated how much we decreased them depending on the baseline respiration. So the higher the respiratory rate initially, the more it would decrease with low-dose rapamycin, suggesting that the mTOR, or at least part of the mTOR pathway and downstream from that, could be involved in this regulation. There's a question, so, we're doing lymphoblastoid cell lines. Is this the case in kiddos also walking around, or is just this a product of the cell lines? So what we did is we looked at kids with autism at their PBMCs, and we put them in the seahorse. And we did indeed find that about 25% of them had this profile where their baseline reserve capacity was elevated and the reserve capacity dropped precipitously with challenge with DMNQ. And you can see that on the left. And then we used a cluster analysis again to cluster these groups, and we found there were 3 groups. This one that was abnormal and 2 others that overlapped with control children in their mitochondrial profiles, suggesting that about a half or third of these kiddos may in fact have this profile. We also asked whether this was different than kiddos with mitochondrial disease, classic mitochondrial disease. So we did the same assay for kids with classic mitochondrial disease, which are the blue circles on the right, and you can see that they did not overlap. So suggesting that this profile is somewhat distinct from classic mitochondrial disease. Recently, we've worked with Dr. Zimmerman and his group to look at some patients who they characterize as having neurodevelopmental regression or developmental regression in autism. And we found that those with neurodevelopmental regression seem to have a different profile in the seahorse with increased mitochondrial respiration for maximal maximal respiratory capacity. And here we see maximal OCR or respiration. And also what's not shown is reserve capacity was also increased in these kiddos with neurodevelopmental regression, suggesting that this may be related to the subgroup of kids with neurodevelopmental regression. So we've come up with this model in the past where we suggest that, well, there's these cells that are under chronic have increased reactive oxygen species, increased oxidative stress, and that some of them have a maladaptive response. And they're like this chronically, and mild acute insults may cause them to lose reserve capacity. So really our theory was, well, maybe, this has happened because of some previous exposure to some type of environmental agent, given that, again, we don't see a lot of genetic disorders to explain these abnormalities in mitochondrial function. And we know that the environment may have a role in autism. So what we did is we exposed these cell lines to DMNQ for a prolonged period of time, for 96 hours, and asked, could this exposure, this prolonged exposure to low levels of oxidative stress, which may simulate how toxicants actually affect the cells, could this change the profile of these cell lines such that they look like those ADAs. And indeed, that's what happened, is that when you take the cell lines and you expose them for prolonged periods of time, 96 hours, to low levels of DMNQ, you actually see then later on, after you remove the DMNQ and such, that the respiratory rates at baseline for reserve capacity and the other parameters increase at baseline, and they're more sensitive to acute DMNQ challenges, suggesting that this is something you can induce. And we did that in the laboratory. So then the question is, does this happen in kids? And so we just published this paper, which we think is really exciting, where we looked at prenatal exposure to air pollution. And so we were able to use zip code information to find out where kids lived, or at least their mothers lived prenatally while they were growing, and derived air pollution exposure data for the kiddos, in which we measured mitochondrial function as children. And what we found is pretty strong correlations, particularly in those kids with neurodevelopmental regression, such that the kiddos' mitochondrial function in childhood was related significantly to the level of PM2.5, which is the measure of air pollution during fetal growth prenatally, while they were so what their mother was exposed to. And there seemed to be different relations. So the relationships were different for those with and without neurodevelopmental regression. Those with regression had a very strong relationship with mostly medium effect size correlations. And then, of course, the question is, well, sure, that's great to look at mitochondrial function. Does this really have any effect on their development and behavior? And then the answer is yes. And what we found is that not only does mitochondrial function affect neurodevelopment and behavior, but that the effect of prenatal air pollution exposure, which is known to be related to autism, that about a quarter of it is that of its effect on development, about a quarter of it is mediated through the mitochondria. That is, the long-term effect of air pollution exposure on the mitochondria influences development. So that's very important, because it gives us a key of where we may be able to intervene to actually affect neurodevelopment in a positive way. And what we did is we used structural equation modeling to look at the nuances of these variables. And so what we showed is that kids with neurodevelopmental regression, for example, neurodevelopment was affected by 3 different factors. It was affected by their exposure to prenatal air pollution. It was affected by their mitochondrial function at the time, and it was affected by redox metabolism at the time. And that mitochondrial function was seemed to be programmed, or at least associated, with their exposure to air pollution early in life. And that behavior was associated with air pollution early in life, and neurodevelopment also. And so the exciting thing here is we're starting to pull out these different aspects of the environment and physiology so that we can intervene to help us understand how to improve the health of kiddos with autism. And kind of the last slide, as we're running out of time, is the fact that we went back and we not only looked at air pollution prenatally, but we used deciduous teeth and looked at metals that the child was exposed to, using an assay that the guys at Mount Sinai School of Medicine have, where they can go through baby teeth and measure the actual exposures prenatally. And what we found, which was very interesting, And this actually goes to, I guess, a little question in the chat there, is that zinc and manganese levels prenatally seem to be associated with long-term changes in mitochondrial function. And what's interesting is this group has really identified zinc, copper, and manganese as very important prenatally as far as increasing the risk of developing autism. And we've linked it now to changes in mitochondrial function long-term. And of course, when you think of those three nutritional metals, superoxide dismutase is really on the top of the list as probably a mechanism. So I won't go through these slides, but I'll thank you for your attention. And if you're interested in this work, we've published 4 papers this year that really talk about this and the possibility of how prenatal influences, both nutritional and other environmental influences, can cause long-term changes in mitochondrial function that may affect development. So the second talk that I'll talk about quickly is our work on looking for treatments for mitochondrial function. So one of the great things, as I said, about metabolic disorders is there's potential that you can actually treat them. I'll just go through these things quickly. So really, there is, I think, somewhat of a controversy in the sense that some people say that it really mitochondrial disorders, only the ones that are primary, that have some type of genetic defect, are the ones that are treatable, and the secondary mitochondrial dysfunction may not be treatable, and they don't treat it. And we published this paper a while back, really led by Dr. Nazoff down in Louisiana, where we argue that it's not the case, that if you find that there is secondary mitochondrial dysfunction, it's really important to treat it. In this paper, I highlight because we actually give examples of the disorders that are clearly not mitochondrial primarily, such as muscular dystrophy, which you find mitochondrial dysfunction in. And these disorders respond to treatment that improves the mitochondria's function. So it really says that, well, it doesn't have to be a primary mitochondrial disorder to treat it. So we know that there's a whole list of different treatments, mostly B vitamins and other cofactors that are important for making the mitochondria work better. And one of these kind of cocktails has been put together in a substance called Neuro Needs, which Dr. Bowles, who we'll be talking about, has really led the development of this compound, which is focused on improving really metabolism in kiddos with autism, with a focus on mitochondrial support. So what we decided to do was to see, well, can we really improve mitochondrial function in kiddos with autism that have mitochondrial dysfunction, and is it related to behavior? So we have a double-blind, placebo-controlled crossover randomized trial that is ongoing right now. And at 3 time points, we're going to measure mitochondrial function. So first we confirm that there's some type of mitochondrial dysfunction. If there is, then they're entered into the trial and we have their baseline mitochondrial function. They receive either placebo or treatment. Of course, we're all blind to which one they receive first. At the end of 12 weeks, we test their mitochondrial function, cognitive function, behavioral symptoms, autism severity, again, from baseline. And then we cross them over to receive either the placebo or treatment and measure this again at the end to determine whether this is there's actually a change. And we're measuring mitochondrial function pretty much non-invasively using the Goldenthal buccal swab at those time points. And our primary aim is to determine if this wide spectrum nutritional supplement can influence mitochondrial function in 50 individuals with autism and abnormal variations in mitochondrial function. That is, they need to have at least one enzymatic measurement that's 2 standard deviations outside of the normal range from the mean, so outside of the normal range, either increased or decreased. And we do hypothesize that that mitochondrial activity will normalize from baseline, and that redox metabolism will improve with this when they have the mitochondrial supplement. We also hypothesize that symptoms on the childhood autism rating scale, on the global clinical global impression severity and improvement, and other measures of behavior, including obsessive compulsive symptoms on the SIBOX, aberrant behavior on the aberrant behavior checklist, anxiety on the parental anxiety scale, caregiver strain, selected parental targeted problems, and general adaptive behavior on the Vineland will improve greater when they're on this mitochondrial treatment as compared to the placebo, and that there will be less complaints of somatic symptoms, including pain, fatigue, and gastrointestinal symptoms. So this is kind of the stages of the study. We developed and organized the study. We have approval. So we went through the process. We asked first for an exemption from the FDA, and they denied us that. So we have a new drug application. We have an IND that is approved by the FDA. We have approval now from the IRB. We spent some time getting a certificate of analysis of the product, as is a very complex product, and so it required actually a special laboratory to be able to differentiate all the components of this and several different techniques, because it ends up when you go to measure individual components, it can be very difficult after they're mixed because they can interfere with each other's assays. We produced an identical placebo, which took some time and went through several layers of and several rounds of taste testing, because this is a powder. So we want to make sure that that it will be that the placebo and the treatment are completely identical. We've advertised and recruited for the study, and we've entered 5 participants so far into the trial, and hope to complete the trial within the next 1.5 years. So with that, I will thank you for your attention. And I think I have 3 minutes for questions if anybody wants to ask questions.
Dr. Frye, there are some questions for you on the chat.
Okay. So let's see. Did you measure superoxide dismutase? Yeah. So we did not. So we are planning to measure oxidative stress, and we use so all our studies in the past, and the study that I had presented, we use the thiol panel developed by Jill James in her lab at Arkansas Children's Hospital, who we work with very closely, and that includes looking at glutathione, both reduced and oxidized, both in the blood and intracellular. So the values that I presented were actually intracellular glutathione redox ratio, that is both the oxidized, reduced, and and the ratio, and other measures there. We don't, though, look at superoxide dismutase, but I would agree that it is important given that it has been shown to be abnormal in kiddos with autism. And as I had mentioned, the nutritional metals that have been implemented really implicate superoxide dismutase. So I think that that is a really good suggestion. Another question, considering that classic mitochondrial testing is not easily available around the world and the less invasive is useful for kids with autism, and we believe so. We've published, I think, three or four papers on the mitochondrial, on the mitoswab. It's not a substitution for a muscle biopsy, but it definitely is extremely useful to consider whether there's a level of mitochondrial dysfunction or even a level of really severe mitochondrial depression that would suggest mitochondrial disease. And yeah, we've published our results as I had shown you, and it has been correlated. So Dr. Goldenthal has a paper where he's validated it against muscle biopsies, and we find that it's very useful not only for detecting a level of mitochondrial dysfunction, but to following along the child to see if you're making a difference with treatment. And then sometimes, usually I'd say most of the time we get, abnormalities that are just outside the normal range in kiddos with autism as far as their enzymatic function. But then you get some kids where complex 1 is very low. It's about 10%, 20%. And these are the kids that usually have some type of genetic disorder or a more classic mitochondrial disease. So I think it's extremely helpful. Yeah.